Human body is the physical substance of the human organism composed of extracellular materials and organized into systems, organs, tissues, and living cells. The basic structural function and biological unit of all known organisms, the smallest unit of life often called the building blocks of life. In the cells we found genes, the basic functional unit of heredity that is found in the tiny spaghetti-like structures called chromosomes. Our body is made up of billions of cells. That is why scientists have taken an interest in the small details in our body to protect us from the diseases such as single gene defect. Have you heard of disease caused by single gene defects? Down syndrome, Marfan syndrome, and AIDS are some examples of it. Scientists have been working for decades on ways to cure disorder caused by mutation genes, and they came up with gene therapy. Gene therapy is a technique that modifies a person's genes to treat or cure diseases. It works in several mechanisms, replacing a disease-causing gene with a healthy copy of the gene. But before anything else, let us know the basics.
What is a gene? Genes are the basic unit of heredity. They contain information about our individual characteristics like eye and hair colors. These are the section of DNA that oversee different functions, which includes making of proteins. Genes came from each our parents, wherein we may inherit their physical traits, as well as mutated genes that can sometimes cause diseases and health conditions. That's when gene therapy comes in.
So how does gene therapy work? Gene therapy replaces a faulty gene or adds a new one to cure and improve your body's ability to fight diseases. In order to insert new genes directly into cells, scientists use a vehicle called vector, which is genetically engineered to deliver the gene. Certain viruses are used as vectors since they can easily deliver the new gene by infecting the cell. But modified viruses cells don't cause diseases. Gene therapy can be used to modify cells inside or outside the body. These methods are in vivo and ex vivo. In vivo gene therapy is done inside the body. A vector will be injected carrying the gene directly into the part of the body that has defective cells. While ex vivo is done outside of the body. Cells are taken out and the vector containing the desired gene is introduced into these cells. The cells are left to multiply in the laboratory and are then injected back into patient, where they continue to multiply and eventually produce the desired effect.
Although gene therapy is a promising treatment option for several diseases, this technique remains risky and under study. To make sure that it will be safe and effective, and when the immune system reaction might happen, the body immune system may see the newly introduced viruses as intruder and attack them. This may cause inflammation and, in severe cases, organ failure. Vector may as well as target the wrong cells. It is possible that the altered viruses may infect additional cells, not just the targeted cells containing the mutated genes. Despite of some possible risks, the treatment option is potentially useful for some inherited disorder like cancer and viral infection. This technique has the ability to cure many of the diseases that have been affected our society for the years.
In the coming future, gene therapy will play an important part in many people's lives. The role of doctors and patients will be changing in the coming years, and it will be due to this therapy.